Okay, so today is Unit 7, Part 2, Protein Synthesis. We've already discussed Part 1 of protein synthesis, which is transcription. Now we're going to talk about translation. Your guiding questions for today are: how is the sequence of amino acids making up a protein determined? And what is the purpose of translation? Make sure to write those down in the top of your Cornell notes. And anytime you see a white question at the top of a slide, be sure to jot that down on the left-hand side of your Cornell notes, and then the notes that follow on the slide or that I give you through lecture, you can jot down to the right of those questions. You should be able to answer both your guiding questions in your journal by the end of the lecture today. Feel free to pause and rewind the video anytime you need. Remember, if you're struggling, write it and have a question, write it down and bring it to class and ask, or come to tutoring. All right, let's get started. So, what is the primary function of a ribosome? Well, remember, the ribosome can be found floating inside the cytoplasm, attached to endoplasmic reticulum, also inside the nucleus, but the primary function of the ribosome is it's the main site of translation. That's not its primary function, though. Its primary function is that it makes proteins. So it is the site of protein synthesis. Ribosomes are referred to as rRNA and act as the site for translation. They're composed of two subunits, large and small, which you can see in this picture right here. This is the larger unit of the ribosome. Here's the smaller unit. This strand right here with the nitrogenous bases written across it is your mRNA. And these little green guys right here that look like Play-Doh, little broccolis, are your tRNA carrying your amino acids. We'll talk more about those here in just a little bit. All right, before I move on this slide, I want you to go ahead and draw and label this diagram of the tRNA anticodon and amino acids in your journal. So tRNA, that's the essential link. Transfer RNA delivers the proper amino acid to the ribosome by reading the mRNA transcript in sets of 3 bases called codons. So you have this codon right here that's on the mRNA. Remember, the mRNA is made in the nucleus and then it leaves the nucleus and brings it to the ribosome. The tRNAs are floating around in the cytoplasm waiting to pick up an amino acid to match up to the codon. The tRNA binds to one of 20 different amino acids at one end, and on the other end of the anticodon, it carries the anticodon, which is the complementary bases to the mRNA codon. Go ahead and pause the video now so that you have time to write any notes down and draw and label your diagram. All right, so the key to the code, what we're looking at here is a codon chart. It says which codons code for which amino acids. Well, if you take a look at this chart and you didn't know what you're looking at first glance, you're like, that's a lot of letters randomly paired out, and you have no idea where to start. Well, on the codon chart, you normally start over on the left-hand side here. Oh, excuse me. Sorry, the left-hand side here. That's your first base. Then up at the top is your second base. Then you come over here and find your third base. So how you do that is you would line up first base, say it's U. Say your second base is a C, so then I'd come down here and highlight this section as a C. And so that carries me just to this box right here. Now I go over here and I'm going to say it's an A, and I highlight that right there. So I have UCA, which is abbreviated for SER for that amino acid. If you notice, I have 1, 2, 3, 4 codons that are different, but they all code for the same amino acid. There are a few that, that code for several. Methionine, this is a special codon. That is our start codon, and that is the AUG is the only one that codes for methionine. These red ones over here are our stop codons, which actually don't count when we talk about the translation, but we'll get into that later. They just tell the ribosome that the transcription or the translation is over. So there are 3 stop codons. All right, translation. So initiation. I'm going to show you a diagram. I want you to be sure to know each part of the complex. I have them labeled here, 1, 2, 3, and 4, and they're also color-coded with the parts of the complex. So that right there is a strand of mRNA. Bringing in the small subunit of the ribosome. Here comes the top part of the ribosome, or the large subunit. The green right here, this is your tRNA, which brings the anticodon to match to the codon, and it brings in the amino acid right here, methionine. So as you can see, tRNA is green, your amino acid, which is attached to the top of that tRNA, is yellow. The anticodon that matches to the mRNA codon is red, and the codon is blue. What I want you guys to remember is a lot of people get confused when we're doing translation, that they use the anticodon on the tRNA to code for that amino acid. That is not the case. You use the mRNA codon to code for the amino acid. Again, AUG is your start codon, and it codes for methionine, which, if you see GAG, blah, 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 CU, the ribosome doesn't start translating until it gets that start codon. Once it gets that start codon, it will go until it gets a stop codon like this, UGA. If you notice, all of these codons are going to code for a different amino acid, and those amino acids are going to link together to form a polypeptide chain or a protein. If you notice, right after the stop codon, there is still a section of the mRNA that is not being translated. The ribosome will continue to read until it gets another start codon, and then it'll translate again. All right, so what happens during elongation? Okay, I want you to use the picture below to help you answer that question in your Cornell notes. Translation or elongation, okay? Right now, I have my mRNA, my ribosome, my tRNAs come in. I've got 1, 2, 3, 4, 5. So it started with the methionine in initiation. Now I've got 1, 2, 3, 4 more amino acids coded. All right, so here comes another tRNA bringing in the next anticodon and amino acid that matches this codon. Comes in, brings it in. So what portion of this complex will eventually become a protein? Looking at this entire complex here, what portion of that will eventually become a protein? Okay. This cycle continues until the stop codon attaches or is read, and then the chain breaks off and goes to do its function. Again, GLY will attach to ARG and continue the polypeptide chain or protein building. Again, remember to feel free to pause the video anytime you need so that you can jot down all the notes you feel are necessary. All right, termination. When a tRNA recognizes one of 3 stop codons, that's what I showed you was in red on the codon chart. proteins called release factors cause the ribosome, the mRNA, and the new polypeptide to dissociate. That means separate. The new polypeptide or protein is now complete. Note the untranslated region beyond the stop codon. Okay, all of this right here just doesn't get translated. Where it shows you 5', 3' end, it's just untranslated. All right, so what is the building block or monomer of polypeptides or proteins? Well, polypeptides, okay, are made up of a string of thousands of amino acids. 20 amino acids are arranged in different orders to make a variety of proteins. If you look at this picture over here to the right, you see several different structures. The names of them are not important. What it is each protein is folded differently. So they're folded in different ways and shapes based on the specific function they need to perform. Okay. Again, this gets into gene expression and how and cell differentiation, where cells and things are made and differentiate for specialized functions. All right. Oh, sorry. Let's go back one more. Nope, I'm good. All right. Amino acids. Amino acids, of course, as we mentioned before, are the building blocks or monomers of proteins. We learned that back in Unit 2, biomolecules. Now, if you're looking at this chart, it might get a little confusing. Let's try to summarize translation as we've gone through it. So what I want you to do right now is just kind of put your pens and pencils down and listen to what I'm saying. Translation is the process of synthesizing or building proteins by linking specific amino acids together in an order determined by the codons found in the mRNA. Remember, codons are sets of 3 specific nucleotides. Each codon designates a specific amino acid except for the 3 stop codons, which cause the newly synthesized polypeptide or protein to be released from the ribosome. Ribosomes are made of ribosomal RNA or rRNA, and in combination with transfer RNA or tRNA and mRNA, catalyze or start translation by allowing the mRNA to be read and the amino acids to be linked together to form a protein. So all of these right here, this, all of these are different structures of amino acids. If you look at ASP and LEU, their structures look very similar, but if you notice, their chemical makeup is different. As in right here, I have a hydrogen bonded to that carbon, but in ASP, there's no hydrogen there. Also, you have carbon and a hydrogen bond here, where you have a nitrogen and a hydrogen bond. So it's small things like that. The order or the arrangement makes them different. Just like the sequence of all these different amino acids makes each protein different. So, we talk about building proteins. Well, why do we need to build proteins? Well, what is a gene? And what do genes have to do with building proteins? Before we start, let's review the central dogma. Everybody should know it by now. We've talked about it several times. So the central dogma is DNA in the nucleus, okay, is transcribed to RNA and is translated to protein on the ribosome. Okay? Before all that can happen, during replication, DNA is copied. So you have replication, DNA to DNA, transcription, DNA to RNA, and translation, RNA to protein. It's very simple. So a gene is a segment of DNA that brings about transcription of a segment of RNA. So gene expression is a highly regulated process DNA contains many regulatory sequences in addition to genes. So your genes can be turned on and off, or expressed or not expressed, just like the light. You can turn a light on and off. It's still there, right? It's just not on. Same thing with genes. So transcription and translation of a gene generally occur when cells need the specific protein or RNA. Cells do not make all possible proteins all of the time. They make them when their body or when they need them to perform a specific function. The purpose of translation is to read the nitrogenous bases of mRNA and convert the triplets of bases or codons, making up the codons, into a specific amino acid sequence, which is the primary structure of a protein. So translation reads the transcript that builds the protein. Now, the purpose of transcription is to produce a protein with a specific function. All right, so now what I want you guys to do is draw a Venn diagram in your journal using the chart here, comparing transcription and translation. Okay, so transcription is a process by which genetic information encoded in DNA is copied onto messenger RNA or mRNA. It occurs in the nucleus, and remember it has to because DNA cannot leave the nucleus, and it's DNA to RNA. Key enzyme complex is RNA polymerase. Translation is a process by which information encoded in mRNA is used to assemble a protein and occurs on a ribosome or rRNA. It's mRNA to a protein or mRNA into a chain of amino acid sequences. It involves tRNA, which brings in the amino acids from the cytoplasm, and amino acids. Go ahead and pause the video so that you can take your time to draw your Venn diagram in your notes. All right, now that you've done that, I'm going to have you pause the video again here in just a second. I've talked to you a little bit about gene regulation, meaning how genes are controlled, how they decide to turn on or off. I want you to pause the video and read and summarize the cartoon here on this slide in your journal. So go ahead, pause the video. When you're done, hit play, and you're almost through. All right, some final notes. In your journal, I want you to write 2 questions that you have over translation and 1 question over a vocabulary term you're struggling with or that you just still don't understand that we can discuss in class on Friday. Be sure to watch the next flip lecture over mutations before coming to class on Tuesday, 1/14. You'll be given the fill-in-the-blank notes for that on Friday. If you have any questions, again, please see Coach Merrifield or Miss Priest for tutoring before or after school, and have a good day.